Oh, wow. Hey, guys. We have got one, two, three, four, five people. And notice it's all women. It's all women. Men are losers. They did not show up. Let's wait a little bit anyway. See if these losers show up. Losers. Anybody have any questions about cardiovascular pharmacology? Is there something I have not answered? Yeah. Is there anything you don't know? Is there anything you don't know about cardiovascular pharmacology? I don't think so. I think you kind of know it all at this point. It's like I've been sitting in this empty room for a whole semester talking to myself. Let's get a chat going. Come on, bring your faces up, people. Oh, wait, this is being recorded. Let me stop that. Is anybody here? Let me see your faces. Oh my God, Celine is here, thank God. There's Nicole. Ariana, you've got a different background. You've gone home. Where do you live, Ariana? You're local, aren't you?
I'm Brooklyn.
Oh, you're in Brooklyn. Oh, I'm totally wrong. Okay. Celine is local. Nicole, where are you from?
Morse Forks.
Morse Forks. I've heard of it.
It's right on the Canadian border.
Oh, okay, okay. And
Like
Noemi,
right on the other side.
Noemi and Rocky, you're both from the city, right? Yep.
Yeah.
By the city, I mean Plattsburgh. Just kidding. Just kidding. All right, so here's the deal, people. We got 2 more classes left, right? Now, in your syllabus, it says we have 4 exams. Doesn't seem right for me to give you an exam on 2 classes. That's going to count as much as all these other exams that counted for everything. Things have just gotten out of control this semester. This Zoom stuff is just, I don't know. So here's what I'm thinking, okay? And I'm looking for your input on this. Rather than having another exam, I had 7 or 8 lectures where I didn't have time to write out notes. I just made PowerPoints out of it, and I gave my lecture based on that. What I want to do is assign each of you one of those lectures and have you transcribe it into notes and send it back to me, and I'll give you 30 bonus points for that. How does that sound? Unless you got an A, then I'm not going to let you do it, because then you don't have to do it.
Quick question.
Yeah?
Do we have to type it out? Because I have notes written out.
I kind of want it typed because then I can use it myself. You know, I'll edit it and then use it myself the next time I do this class.
Wow, you're so picky.
Well, I'm trying to get you to do my work for me. That's
I see that.
what leadership is all about.
I think that's a good idea.
You think it's a good idea?
I agree.
I think it's going to take the pressure off this week and still give you a chance to improve your grade. So
When would that be due?
I'd like it due Thursday, but I mean, next week is finals week. You get it in next week if you have to.
Okay.
So I think you're going to have more time this week than next week because you'll have to study for other finals. So I'll make it due Thursday, but you know, it's an open deadline.
All right.
Okay. And again, if you got an A in the class, I won't ask you to do it because there's no point at that point. All right. Okay. Well, I put together a lecture today, and you're not going to be tested on it. But what the hell? As long as we got the lecture, let's go ahead and do it. All right. So I don't know if you had a chance to read the paper. I think Ariana did because she gave me some info on it, which was kind of nice of her. Was it Ariana? No? Who? Celine? Someone sent me some info on this. Hang on a sec. I don't know. Oh no, it was our Ethiopian friend. What is her name? I can't believe I'm blanking on this. Oh my God. I'm an idiot. All right, wait, I'm going to come up with their name if it's the last thing I do. It could be. Bezowit. Bezowit. I always pronounce it wrong. Bezowit. Okay. So that's neither here nor there. Let's do this lecture. Serelexin. So serelexin, what I liked about this paper was it connected. It's a hormone I've never heard of before, but it made a lot of sense when I read what this hormone is supposed to do, just based on my own personal experiences. So serelexin. Do I have a? Relaxin is the hormone. Serelexin is the recombinant form that's made as a drug. So increases cardiac output, renal blood flow, arterial compliance, relaxes pelvic ligaments, and softens pubic symphysis. So the last three is what kind of lines up with my experiences. So 2001, I was doing a sabbatical at the University of UVM's College of Pharmacology, and I was working on cerebrovascular arteries. So I'd take a rat, kill the rat, open the head up, and take this major artery that comes in through the back. And then I take those arteries and incubate them in proteases and digest all of the extracellular proteins, and then it would fall apart into cells. And then I would take those individual cells and poke an electrode in them and do whole cell electrophysiology with them. It was a lot of fun. But let's see, I started in July, and by November, I still had almost no data, because sometimes the cells were good, but mostly the cells were bad. And it's like the cells were just falling apart, and they just were horrible cells. But, just so I didn't have to kill a rat every day, I'd find someone in the department who was already killing a rat for some other reason, and I just take the head. And there was someone in the department who was using pregnant rats. And so some days I'd have a male rat, sometimes I'd have a normal female rat, and sometimes I'd have a pregnant female rat. And it turned out that it always worked the best when I had a pregnant female rat. The cells would always come out really nice, but all the other rats, they were terrible. And I figured out after a while what was going on was the pregnant female rats didn't have as much extracellular matrix, and the cells came apart easier, and they were nicer. And so I figured out that if I had a female rat, a pregnant female rat, I'd have to incubate for 10 minutes. A female rat was 15 minutes, and a male rat, 20 minutes in the proteases to get the cells to fall apart. And then after I figured that out, I got great data for the next 6 months, got a paper out of it. But it was that piece of information there. Why is it that female rats have different vasculature? I mean, pregnant rats, their vasculature was a lot softer. Excuse me. And then years later, got married, had a kid, and my wife had a lot of migraine problems before she got pregnant. But then after she had 2 kids, her migraine problems pretty much went away. So it seems like it changed the vasculature to her head somehow, although that's just speculative. That's just the way it seems to me. So I'm like, maybe there's something about this. And now I'm reading about this hormone, and that's what it does, right? It changes arterial compliance, the elasticity. And also, important for pregnancy, relaxes the pelvic ligaments so that the hips and everything get looser, and softens the pubic symphysis, the connection between the 2 pelvic bones, relaxes them, all of it just to loosen up the pelvis so that it can open more to allow the child out during pregnancy. So that's why I kind of liked this paper, because it brought up this interesting hormone I'd never heard about before that kind of connected those two aspects of my experiences. What does relaxin do? It's got a G protein coupled receptor mechanism. Why did I say that without saying what it was linked to? I don't know. I don't know if it's IP3 or cyclic AMP, one or the other. I would say probably IP3, because number 2, it says activates the nitric oxide pathway. And we know that's activated by calcium, calmodulin, nitric oxide synthase, that pathway. So that's going to cause relaxation, right? Nitric oxide is a relaxer. And it releases vascular endothelial growth factor and matrix metalloproteinases. So that's going to increase vascular, that's going to make vascular changes too, right? VEGF causes endothelial cells to divide so you can, get more vasculature. And your MMPs break down extracellular matrix, which again gives you more compliance, more elasticity, less stiffness from the arteries, and that's kind of what I was talking about with the pregnant rats. But I think I said this. Oh yes, I did. This is released during pregnancy, and through all of this, it remodels arteries. Remodel is a word they use a lot in cardiovascular science. It just means changes. It changes the way the arteries are laid out. So relaxin, interesting hormone. Serelexin then, it's just a recombinant form. So you take the messenger RNA, you make DNA, and you make the protein from it. And this you can administer by IV. And it's been shown to have some clinical benefits in acute heart failure. So in dyspnea relief, which is shortness of breath, reduced in-hospital worsening of heart failure. So it decreased mortality in 180 days. So it seems to help with heart failure. And why is that? The current study, they're looking at what effects it might have on coronary artery disease. It has effects on endothelial dysfunction, oxidative stress, and inflammation. So coronary artery disease, the way that develops, as we talked about before, was the endothelial cells don't work as well and they separate, and then you get LDLs going into that subendothelial space, and the cholesterol gets oxidized, and then that causes inflammation, and that's kind of like the beginning of the whole coronary artery disease sequence. So if it can inhibit this and can have some good effects on coronary artery disease. And so what they're trying to do is determine what is the effect of this on vascular oxidative stress, endothelial function, and atherosclerotic lesion formation. So is this going to be able to inhibit atherosclerotic lesions? And the model that they're using, whole animal model, is mice that are deficient in apolipoprotein protein E. So basically both copies of the gene for ApoE are deleted in this mouse model. And ApoE is the protein on LDL that binds to the LDL receptor. So if it doesn't have that protein, then LDL just hangs out in the blood, and it becomes like a familial hypercholesterolemia model. Most familial hypercholesterolemia is a lack of the LDL receptor, but this will also cause the same dysfunction. So these mice are known to develop coronary artery disease at a young age and die younger than normal mice. Okay, so, excuse me. First thing they did is their entire study is going to be over 6 weeks. So they want to make sure that serelexin itself is not going to change important cardiovascular parameters over that 6-week period, right? Because that would be a confounding factor. You think the results is because of this, but it's actually because of that. So you want to make sure that the only changes you see are the things that you're looking at. So they treated these mice, these ApoE negative mice, for 6 weeks with vehicle, which is just sodium acetate, serelexin at 0.05 micrograms per hour, and then serelexin at double that amount. And they look at total cholesterol. Oh, and by the way, they're on a very high cholesterol, high fat diet through this whole experiment. So normal mice then, serelexin 2, no change in cholesterol, no real change in body weight, no change in systolic blood pressure, fasting glucose is the same, and heart rate is the same for all of them. So serelexin didn't cause any changes in anything, any of these parameters. So that was just kind of like a negative control. You want to make sure none of that stuff is affected. Here's how the experiment actually went. You take these ApoE negative mice and treat them for 2 weeks in a high-fat, cholesterol-rich diet. Now, at that point, you take all your mice and you split them into 3 different groups randomly. One group gets a vehicle mini pump implanted. I'll talk about the mini pump in a second, but basically they're getting nothing but sodium acetate for 4 weeks. And then another group gets 0.05 of serelexin for 4 weeks, and the other one gets 0.1 for 4 weeks. So at the day 14 of the experiment, which is when it actually starts, they put one of these osmotic mini pumps in, which is a really cool little mechanism. I think they have these for humans too, but the little bit of research I did showed it was mostly for animal models. But anyway, this is like a capsule, and you can see it's about this big, so it sticks out on a mouse, although you can imagine it wouldn't even be noticed on a human. But you've got an outside layer here, which is permeable to water. And then inside of that, you've got an osmotic layer. So basically high concentration of salt in a gel or something like that. So it's sucking water in. And then you've got an impermeable membrane here. And then you've got your drug inside that impermeable membrane. And over at this end, you've got like a nozzle that's controlling flow out of this reservoir. So basically, you set this inside the mouse, and the osmotic pressure pulls water in. And as it does, this osmotic layer swells and it puts pressure on the impermeable reservoir and squeezes that reservoir. So it's putting pressure on the reservoir. And the release from the reservoir is being controlled by that flow moderator. So it's a really neat way. If you want a constant infusion in a mouse, you can't put it in a hospital bed and hook it up to an IV. This is a nice way to get a constant infusion experimentally. So I think it's a really neat little model. So anyway, what happens? So first thing you want to do is show, did you actually get the concentration of serelexin that you expect or you hoped for in the mouse? So here is vehicle, and of course there's none because it's exogenous. It's not something the mouse would normally be expressing. And then here's a 0.5, gives you a concentration of this. What is this? Plasma level in nanograms per mil. So it gives you about 10 nanograms per mil. You double the dose, and the concentration you get is almost double. So it does seem to be working. The mini pump is working. You're getting the serelexin in there. Then the next thing they're looking at is superoxide production. So that was one of the things we were hoping to inhibit, because that oxidation is part of the process of cholesterol being oxidized and starting the atherosclerotic plaque. So here with just the vehicle, you get about this much superoxide production. Oops, shoot. And then with serelexin, at either dose, you're getting a dramatic decrease in the amount of superoxide production. So that's great. It seems to be inhibiting production of superoxides. Number two, the second thing they want to talk about is endothelial dysfunction. So this is based in a mechanism that we talked about early in the semester. This is one of the Furchtgott series of lectures. So here we had, the way this works is, and I used to do these experiments myself, like when I used to do aortic ring contractions, this was always one of the first experiments I did just to test that the endothelium was okay. You get your ring in the bath, you contract it with norepinephrine, and then you hit it with carbachol. And if your endothelium is intact, it'll relax almost to nothing, almost totally back down to baseline. And that's happening because of nitric oxide mechanism. Your carbachol is a muscarinic agonist. So it activates the muscarinic receptor in the endothelial cell. That activates production of IP3, release of calcium, calcium binds calmodulin, activates nitric oxide synthase, production of nitric oxide, which diffuses over into the smooth muscle cell, activates guanyl cyclase, and that produces cyclic GMP. And cyclic G then activates G kinase, and that's got, I think, 5 different targets in the cell that cause relaxation. So that's the carbachol. That's testing for endothelial cell function. Now if you don't get a response with carbachol, two possibilities. One is your endothelial cells aren't good. The other is your smooth muscle cells don't have any guanyl cyclase. So to tell the difference between those two possibilities, you test with nitroglycerin, because nitroglycerin will go into that smooth muscle cell and directly cause relaxation by production of cyclic GMP. So we're looking at carbachol versus nitroglycerin. NTG is just a smooth muscle cell. Carbachol is looking for endothelial cell function. And here's what you see. In the vehicle, you don't get that much relaxation with carbachol. This is a dose response curve, carbachol concentration versus vasodilation or relaxation. So with the normal ones, you get at most 75% relaxation. With the vehicle treated, you get good relaxation, all the way down to, looks like 30 or 15% contraction left after the relaxation. So this serelexin seems to have restored the endothelial cell function. And here you can see with the black line that in the normal vehicle treated ones, their cyclic GMP function is fine. The smooth muscle cells are fine. So the big difference then is that the endothelial cells are not functioning well. And again, this is probably, they didn't look at this directly, but probably because, you've got high LDL and that's infiltrating under the endothelial cells and causing inflammation, and the endothelial cells are not working properly. Of course, they didn't look at that directly, but that's probably what's going on here. Okay, what they did look at directly, though, is plaque buildup over that 4-week period, or well, actually over the whole 6-week period. So here is vehicle, the red is plaque, okay, and here is serelexin. You can see maybe a little less plaque, hard to tell. I mean, they look pretty similar. Here's a higher concentration of serelexin. Now you can see large areas that don't have plaque on them. So it does look like there's less plaque here than here, but you don't have to go by what it looks like. They've done some measurements here. So this is a measure of lesion severity, like how bad are the lesions. And I didn't read the methods close enough to see how they would determine that, but you can see, you see a trend in the direction that you're hoping, but none of this data is statistically significant, so you can't really say much about that. And then also over here, this is they're measuring the difference between early plaques versus late plaques. So what percentage of the plaques in the arteries look like they're just starting, and what ones look like they're well established. So in the plaques in the serelexin treated mice, seems like there are more early than there are in the late, and there are fewer late than there are in the vehicle. So it looks like it's moving in the direction you would hope, but again, this is not statistically significant, so you can't really say anything about that. You can see the amount of variation they get. And that's always a problem when you're working with whole animals. If you can take cells in culture and work with them, you've got very few, you've eliminated a lot of the variables you got to deal with, so you can get a lot better data. With a whole animal, you're going to have a lot of variation, so you need a lot of animals to get that standard deviation down to make things statistically relevant. But it looks like things are going in the right direction, but again, you really can't say that because of it's not statistically significant. Now here, this is one that is significant, which is the absolute size of the plaque in millimeters. And you can see you do see a decrease in the size of plaques with the serelexin. And percentage of the artery wall covered with plaque also decreases with the high dose of serelexin. Looks like it's decreasing with the low dose, but again, it's not statistically significant. So we are getting some protection from plaque development. Collagen buildup. So the atheroma, part of that, once you get infiltration, inflammation, smooth muscle cells start to grow out, and you get collagen being put out into that atheroma, you're getting collagen buildup there. So here with collagen buildup, you're not really seeing any difference with serelexin. Macrophage infiltration, again, not much difference. And now we're going to look at other white blood cell infiltration. You're not seeing much difference, no real difference in inflammation. So it's not directly causing any changes in inflammation, and it's not changing the vascular smooth muscle cell infiltration either. So we're not seeing much changes in that. It seems like it's working more on the earlier parts of the atheroma development, the oxidation of cholesterol and the endothelial cell dysfunction part of it. Okay, and then they did quantitative PCR analysis of specific things that are supposed to come up or down, things that are supposed to change with serelexin exposure. So number one, this is the angiotensin II receptor, which goes down with serelexin, statistically significant in this case. This is the endothelin receptor. Endothelin is a relaxin. I believe it's a hormone released by the endothelium that I believe causes relaxation of the smooth muscle. And so this receptor goes up with serelexin, not significantly though. No change really in nitric oxide synthase, and no change in the VEGF. So we said that serelexin usually is supposed to cause production of vascular endothelial growth factor. Here they're not seeing any of that. You can see that's about as insignificant as you can get. And this is not significant. And the only thing that's significant is the upregulation of the angiotensin II receptor. Although it says not statistically significant, but you can see PO2, usually PO5 is significant, but anyway. And inflammatory mediators that are found in the plasma. So if these things are happening at the arterial cell wall level, they're going to end up in the plasma. So they're detecting inflammatory hormone, inflammatory and anti-inflammatory hormones to see if they go up with this treatment. And here with cytokine IL-10, which is anti-inflammatory, you can see it does go up. And with the pro-inflammatory cytokine IL-6, it goes down. So, that helps to show us that we're not getting that's some markers that inflammation is being decreased in the whole mouse, which hopefully means that you're getting less atheroma development because of that. Okay, discussion. So what can we actually say about this data? The thing about the paper is they saw some of the things that they wanted to see, they're hoping to see. They didn't see some of the other things. So they have to limit their conclusions based on what was statistically significant. They did see a reduction in vascular oxidative stress. So that was superoxide production in the vasculature itself. It reversed the endothelial dysfunction. The endothelial cells were working much better, reduced the atherosclerotic plaque development, and trended towards, they said, which is kind of saying, that's kind of like BSing a little bit, trended towards reduction of plaque severity, because it really didn't statistically, but it looked like it was going in that direction. No change in the collagen, smooth muscle cell content, macrophage, T cell, or neutrophil infiltration. So they saw no changes in those things that they were hoping to see. And they did get an upregulation of, wait. No, I got this backwards. Shoot. Oh. Escape. Down regulation of pro-inflammatory and up regulation of anti-inflammatory. Okay, so that was a good effect that they did see. Okay, reduction in atheroplaque size, that was significant without an effect on collagen or vascular smooth muscle content. So why is it that they saw, slight differences, but not as much as they want? One thing what they thought was maybe it's kind of a short period, 4 weeks. When you think about coronary artery disease, it develops in humans, it probably starts in the 20s and it starts to cause problems in the 50s. And you figure if you normally would live 80 years, that would be 30 out of 80 years, that's 3/8 of your life. How long does a mouse live? Usually 2 years. So is this really a relevant time span? It's probably not enough. They probably should do it longer. And also, it seemed to be dose dependent at 0.05 mgs per second, or microgram per second, or whatever the dose rate was. Seemed to have certain effects, but the effects went up when you went to the higher dose. So that's it, and that's all. That's why they think they didn't see what they wanted. So kind of an interesting paper. And it's kind of shows why, to a certain extent, it's like another reason before I left, I said I was going to give you 2 papers a day for the last two weeks. And I'm having a hard time finding good papers because so much of this cardiovascular stuff, the important work was done 60s, 70s, 80s, and even into the 90s. But now it's like kind of chipping away at the edges, but there's nothing really exciting going on right now. I think all the real exciting research now is in cell division and cancer therapies and things. And obviously COVID research, which is kind of big. By the way, I don't know if I told you this, but COVID gets into cells via the angiotensin II receptor, which is kind of an interesting factoid. So it kind of dovetails with what we're talking about. So let me bring your faces up again real quick. Except Noemi, I know you're embarrassed about your hair net, but that's fine. Doesn't bother us. Okay, what time is it anyway? 10:04. So a short thing today. Thursday, why don't we get together Thursday? Do we need to get together Thursday? Let's get together. I miss you guys when you're not around. Why don't we meet Thursday and we can talk or something, or we'll do something, just because I think legally we're required to get together. But I'll send you that assignment, and hopefully you get that to me by Thursday, if not by next week, and then that will be our class. Okay, everybody happy? Bye bye. See you later.